And now there is no delay. So we'll start our lecture. Over to you, Alisa. Please carry on.
Yes, thank you so much for this enlightening introduction about me. Just I am trying actually to share the presentation here screen so that I can start. I'm not seeing any options here to open the presentation.
You can see share screen button on your beside chart. So you can
Yes.
hit that button, share screen. I hope you got it.
Yes.
Did you get it, sir?
Yeah, I am opening it. Yes, is the presentation visible on screen?
Yeah, it's visible.
Okay.
Please carry on.
Yes, thank you so much. I regret for the delay. Yes, before I start, I would like to discuss my current position. Right now, I am working associated with Green Lab. Green Lab is a concept from the Alforage Industries. They are planning to open a new lab for the diagnosis of food, water, pharma, and cosmetics. And I have given the role to set up the microbiology section of this laboratory. Basically, I'm specialized in microbiology, so most of this food diagnosis part, we will be covering microbiology diagnosis. Of course, I will give some idea about the chemical analysis, but by the major focus will be on the microbial analysis of food and water. Before working in Green Lab, I was associated with the Riyadh Municipality Central Area Labs. It is a central lab for the entire city, capital city for Saudi Arabia. And I got nice experience to set up the microbiology lab in this municipality for I worked there for 3 to 4 years, and now I am associated with Green Lab. So the today's my topic, diagnosis of food and water as a quality perspective. Here I am mainly focusing the diagnosis with respect to human health. Generally, we can diagnose food and water for various pathogens. But according to the quality perspective, according to the human health point of view, what is the type of diagnosis we have to make? So this is the main focus of this presentation, because if we want to diagnose any food pathogen or the water contamination or the pathogenic bacteria present in water, we have number of tests. But which is the most important one for the human health point of view and for the quality, that is the main focus here. So let us begin. Before we go into the details, let us see some of the definitions given as per ISO 22000, because here the definition of food is different than the general definition. According to the ISO 22000, food is a substance or any ingredient which is processed completely or semi-processed or raw form, and it is intended for consumption, and it includes drinks, chewing gum, any substance which has been used in the manufacture preparation or treatment of food. So not only the complete food, but the semi-processed food or the raw material or the ingredients, these all are coming under the category of food. Food does not include cosmetics or tobacco. So excluding this or any substances which are used for drugs. Food is intended for consumption by humans and animals and includes feed and animal food. So this definition of food also includes the feed which is used for feeding the animals. Feed is intended to be fed to food producing animals. Food producing animals like farm animals. Animal food is intended to be fed to non-food producing animals such as pets. So these all categories are coming under food. And animal food. The definition for animal food: single or multiple products, whether processed, semi-processed or raw, which are intended to be fed to non-food producing animals, are coming under the category of animal food. Also, the food is intended for consumption by humans and animals and includes feed and animal food. So these are some terms as per the ISO 22000. Then this is the food chain. The food chain, according to ISO 22000, is involving different stages. The sequence of stages includes production, processing, distribution, storage, handling of food, and also the handling of ingredients of food. Broadly, from the time of primary production till the consumption, all these stages are coming under food chain. And this includes production of feed and animal food, production of materials intended to come into contact with the food of raw materials, and the service providers. So this is a different food chain which generally we study academically, but this is according to ISO 22000. Then regarding the food safety, what is food safety? Food safety is the assurance that food will not cause an adverse health effect for the consumer when it is prepared or consumed in accordance with its intended use. Food safety is related to occurrence of food safety hazards in end products and does not include other health aspects related, for example, malnutrition. So malnutrition should not be confused with the food safety. It is a different aspect. And it is not to be confused with the availability of the food or food security. This includes feed and animal food. Then what is food safety hazard? This is our main focus for today's presentation. Biological, chemical or physical agent in the food with the potential to cause an adverse health effect is called food safety hazard. Here, the term hazard should not be confused with the term risk, which in the context of food means a function of probability of an adverse health effect. For example, becoming disease and the severity of that effect, example, death. So this should not be confused with the hazard. Then coming to the quality, there are three aspects of food quality: safety, authenticity, and nutrition. Broadly, when we do diagnosis, we have two parts: microbial analysis and chemical analysis. Chemical analysis mainly focuses on nutritive quality of the food and also the authenticity of the food. If the label the claim label on the food, we can do by chemical analysis. And microbiology analysis is giving the safety of the food, whether it is safe to consume by the human body or not. And you can see there is one authenticity I have slightly highlighted in a different color for microbial analysis, because in some part we can do authenticity by microbial analysis also. But majorly the authenticity of the food, for example, meat adulteration, we can do by chemical analysis. Then food poisoning, according to the US FDA, the foodborne illness or foodborne disease, it is also referred as food poisoning, is any illness resulting from food spoilage. So whenever the food is undergoing spoilage, it will result into food illness. Broadly, this is because of pathogenic bacteria, yeast and mold, parasites that contaminate food, viruses, toxins, and chemicals. So toxins can also lead to food illness or food poisoning and certain chemicals. Causes of food poisoning as per US FDA. US FDA has classified 8 causes for food poisoning. The number 1 is bacteria. These are very common cause of foodborne illness. Number 2, enterotoxins. These are protein exotoxins that targets our intestine. Enterotoxins can produce illness even when the microbes that produce them have been killed. This is very important because sometimes we feel that cooking of the food is enough, but there are some enterotoxins which are heat stable. So that's why it is very important to test for enterotoxins in the food. So as they are heat stable. So even though the producing organism is killed during the process of cooking, but the toxins are still present, which can lead to food poisoning. What are the categories of these enterotoxins? Broadly, they are produced by 3 different organisms. Staphylococcus is producing enterotoxin A and B. Clostridium botulinum and Clostridium perfringens are producing enterotoxin, and Bacillus cereus is also producing toxins. Out of these 3 types of bacteria, Clostridium are the anaerobic organisms, whereas Staphylococcus and Bacillus cereus are aerobic organisms. Then the number 3rd cause of food poisoning is yeast and mold. Here, for the food diagnosis, there is a test called total count of yeast and mold. So according to the US FDA, we have to see the general count of yeast and mold. There is no special species diagnosed tested here, Candida albicans or Aspergillus niger or Penicillium. It is a total yeast and mold count because there is certain amount fixed which can be tolerated by the immune system of our body. Based on this, the US FDA has given the guidelines. What are the permissible level of yeast and mold? So it's a general test. Then viruses can also cause poisoning, especially the norovirus is causing the problem in water pollution, water contamination. Chemicals like benzene, dioxins, polychlorinated biphenyls, perchlorate, PFAS, radionuclides, these all chemicals can lead to food poisoning. Metals like arsenic, lead, mercury can lead to food poisoning. Pesticides, mycotoxins like aflatoxins, patulin, ochratoxins, especially aflatoxins are more dangerous as they are cancer-causing agents. So these are different causes, different type of organisms which can cause food poisoning. This is the importance of the foodborne illness every year. These are some reports from the Saudi Arabia and from the America, from the CDC. While the American food supply is among the safest in the world, we all know, but the federal government estimates that there are about 48 million cases of foodborne illness every year. This estimate is equivalent to 1 in 6 Americans becoming sick from contaminated food, which results in an estimated 128,000 hospitalizations and 3,000 deaths. This is the data taken from CDC. There is a report by the Saudi Ministry of Health revealing that 2,191 cases of food poisoning were recorded during 2018, and out of which 3 people died. While the estimated annual general rate of death due to food poisoning is 1 lakh people in the kingdom. Let us go for the importance of food microbiology laboratory. As I am a food microbiologist, I will be focusing mainly on the food diagnosis with respect to microbiology. Food testing laboratory forms the backbone of foodborne detection. The function of the laboratory is by detecting clusters of related diseases, the disease cases that may later be identified as outbreak. So initially, the laboratory is diagnosing the presence of a dangerous organism in the food, which in the later stages will be identified as an outbreak. Routine accurate testing is the only way to identify harmful food contaminants and adulterants. So we have to routinely test the food for the adulterants and contaminants. Then only we can find out the outbreaks. Public health laboratory partners with the government municipality, without which many widespread foodborne outbreaks would not be detected. So the laboratories are going in hand with the government municipalities, and they do routine testing, and the results are shared with the government. Food testing laboratories perform specialized testing on thousands of foodborne diseases isolates from variety of marketplaces. So the food testing laboratory is taking the samples from the restaurant, bakeries, hotels, all the food area. Epidemiologists, regulators, and policymakers use the outcome of the laboratory analysis reports. So whatever the reports generate from the laboratory, these are used by the policymakers and epidemiologists to make new policies for the safety of the human health. These are the list of the commonly foodborne bacteria, and on the another side we can see the less common foodborne bacteria. The common foodborne bacteria which can cause food poisoning, there are 14 I have listed. The list is according to US FDA. Campylobacter jejuni, Clostridium perfringens, Salmonella species, E. coli O1Y7 and H7. This E. coli species is different from the normal E. coli. This is a toxin producing E. coli. So, and it is very dangerous. Only the 10 cells, if they enter in our body, can cause severe poisoning leading to death. So this is very dangerous bacteria, E. coli 157H7. Bacillus cereus, Listeria monocytogenes, Shigella species, Staphylococcus aureus, Staphylococcus enteritis, Streptococcus, Vibrio cholerae, including oven and non-oven species, Vibrio parahemolyticus, Vibrio fulnificus. And the last one, Yersinia enterocolitica and Yersinia pseudotuberculosis. These are the common bacteria responsible for the food poisoning. But the other less common bacteria includes Brucella species, Corynebacterium ulcerans, Coxiella burnetii causing pew fever, and Plasmonas schizoloides. So this is the reason I was telling in the beginning that when we do food diagnosis, we can test for variety of number of bacteria, but the main focus for the human health point of view is these 14 pathogens. These are the waterborne pathogens. The pathogens which are responsible for spoiling the water and leading to infections are coliforms, E. coli, Pseudomonas aeruginosa, fecal streptococci, and thermotolerant coliforms. The less common waterborne bacteria includes Legionella pneumophila, sulfide-reducing Clostridia, and heterotrophic bacteria. These are the foodborne diseases. Some examples I am discussing here. For example, Salmonella is causing salmonellosis. It is commonly contaminating meat, poultry, milk and dairy products, egg, seafood, and fruits and vegetables. As I am associated in a Gulf country, Saudi Arabia, here for the food testing by the Gulf standards and by the Saudi Arabian standards, Salmonella is needed to be tested in every type of food sample, whether it is a fruit, vegetable, food, cooked food, rice, okay, the water, not water, sorry, all the types of foods, we have to test the Salmonella here, and the permissible level is 0 here, because all the species of Salmonella are very pathogenic. So even there is one bacteria detected Salmonella, we have to reject the sample. Then coming to Campylobacter, the most important species here is Campylobacter jejuni. It is a organism which is it is a microaerophile, needing a small amount of oxygen and carbon dioxide. It is a very tricky organism to grow in lab, needs special instruments to cultivate this organism. It is commonly contaminating foods, including undercooked raw poultry, dairy products, non-chlorinated water, sausage, meat, shellfish, fruits, and mushrooms. Normally, we test Campylobacter in egg and in chicken samples. Coming to Listeria, the important species here is monocytogenes. It is responsible for causing listeriosis. The foods which are commonly contaminated by Listeria includes uncooked meat, uncooked vegetables, and processed food, hot dogs, smoked seafood, unpasteurized raw milk, and cheese. Especially for dairy products, Listeria has to be tested. E. coli, we all know it is an indicator of fecal contamination in water. So it is the most important test for the sanitary quality of water. Food as a vector of transmission, including underground beef, unpasteurized milk. Here, the E. coli not only is tested in water, but also in the food samples. And one more thing, E. coli is also tested for hygiene point of view in the worker hands, and the swabs taken from the meat, and the swabs taken from the kitchen, from the restaurant. So E. coli is a very important indicator for the sanitary quality, not only for the water, but for the food processing and food preparation areas. Shigella, causing shigellosis, is found in polluted water and feces. It contaminates salads, potato, tuna, shrimp, raw vegetables, milk and dairy products, and poultry. Here, this organism we are not testing here. Generally, the test is done in English countries. Staphylococcus aureus causes staphylococcus food poisoning. It contaminates foods, including meats, meat products, poultry, egg products, salads, bakery products, sandwiches, milk and dairy products. Then Clostridium. Clostridium is commonly producing toxins. So it is contaminating cooked meat, poultry, non-refrigerated cooked meat, non-refrigerated cooked meat, and poultry. Here we also do Clostridium in canned food. This is a very important test for the canned food. Clostridium perfringens, we have to test, and the procedure of testing is very tedious. The first step of the diagnosis is taking 10 days for Clostridium. Vibrio is causing cholera. It contaminates foods including fish, seafoods like prawns, tuna, and oyster. Bacillus cereus is mainly responsible for diarrheal food poisoning. For a moment, I want to hold here. My voice is audible?
Yeah,
Hello?
please continue,
Yes,
sir.
yes, sir.
Yes.
It is
Okay.
audible.
It's audible. Please continue.
Yes. Bacillus cereus is responsible for diarrheal food poisoning, and it is contaminating meat, processed meat, stew, spice, soups, vegetables, and processed food. Now, these are some of the organisms which are not commonly tested in Saudi Arabia and Gulf countries, but the Western countries are testing these organisms. Shigella, Yersinia enterocolitica, another species of Yersinia pseudotuberculosis, Vibrio cholera, Clostridium botulinum, Legionella pneumophila. Parasitic animals like Cyclospora, Cryptosporidium, and viruses like hepatitis A and norovirus is commonly tested in water samples. Foods from genetically engineered plants like potatoes, apples, squash, papaya, soya bean, corn. Here in Saudi Arabia, genetically modified products are banned. So I don't know, but maybe in India they are testing by some guidelines given by FSSAI. But here for the Gulf countries, especially Saudi Arabia, the genetically engineered products are banned here. So we are not testing this. Ingredients from genetically engineered plants: cornstarch, corn syrup, corn oil, salad dressings, sugar from sugar beets. So these are the other tests which are done in the English countries. These are these tests which are done in the Gulf countries for microbiology diagnosis of food. These are 15 pathogens here. And there are two general tests: aerobic plate count and yeast and mold. E. coli, Bacillus cereus, Staph aureus, Enterobacteriaceae family, Listeria monocytogenes, E. coli 157 and H7, Salmonella species, Campylobacter jejuni, Vibrio, Clostridium, Chronobacter sakazaki. Here I would like to point out Chronobacter sakazaki. This bacteria is responsible for spoilage of infant food. The formula milk and the Cerelac, the Nestle products, especially targeted for infant diet, are generally contaminated by Chronobacter sakazaki. So any infant food sample is given, we have to test for Chronobacter sakazaki. Pseudomonas, fecal streptococci, coliform, sulfide-reducing Clostridia. These four are tested in water analysis. Yes, now regarding the methodology followed for the processing, there are two types of standards here. One standard is for the method of analysis, and another standard is the limits, the permissible level for each organism in the food sample. For processing point of view, we are following the US FDA, there is a bacteriological analytical manual called as BORNE. BORNE is giving all procedures for the diagnosis of food and water related to microbiology. And the next one is ISO standard. For Gulf countries, we have GSO, and for Saudi Arabia specific, we have SASO standards. And for food analysis, for water analysis, we follow APHA, American Public Health Association. In India, they follow FSSAI and also the ISI standards. Currently, I am following ISO and GSO standards here for food analysis. And for water analysis, I am following APHA in our laboratory. Most of the laboratories here in Saudi Arabia, they follow ISO and GSO standards. Now, coming towards the limits, the limits for each type of sample, how many bacteria present should be allowed in a sample, this is given by Codex Alimentarius. It is a main standard. Based on the Codex Alimentarius, different countries have standardized their limits according to their preferences. The GSO, I am following presently GSO limits. I will be sharing one screenshot from the GSO limits for criteria for testing microorganisms in the food samples. Then the WHO is also giving limits, the permissible limits for different types of food and water samples. And sometimes European and other standards are considered when there is no availability of any sample, any matrix, we are not finding limits, we can refer for European and other standards. This is the screenshot for the GSO standard, which I'm currently using. This is implemented from 2015. The code number is GSO 1016. Here, I want to highlight each item of the food. For example, the raw meat, including chilled and frozen, or whole or half carcass pieces. The test which the microbiology lab has to do, there are 3 tests here: aerobic plate count, Salmonella, E. coli, O157. Out of these 3, aerobic plate count is the enumeration test, whereas the Salmonella and E. coli are detection tests. In the coming slides, I will discuss the difference between these two, the enumeration and detection. Here you can see on the right side, the guidance, the number of samples to be tested, what is the permissible level, M and capital M. So we have to test 5 samples for each category, and each sample will be replicated. And these are the permissible level, 10 to the power of 5. If any sample exceeding 10 to the power of 5 should be rejected. So how many samples out of 5 samples, if 2 are having 10 to the power of more than 10 to the power of 5, they will be rejected. Similarly, you can see fresh poultry, raw meats. This is number 3 is for one type of food, meat, poultry, and its products. Similarly, we have the GSO standard for all the foods, for dairy products, for bakery products, for milk, for ready-to-eat foods, for water samples. Now, coming to the main part of diagnosis. The diagnostic tests in microbiology broadly are of 2 types: enumeration test and detection test. Enumeration test, we have to count the number of organisms. Under the enumeration test, we have 2 categories: general test and specific test. General test is the total aerobic count of the bacteria and total count of yeast and mold. For a specific enumeration test, we have to enumerate the number of Staphylococcus aureus in the sample. For Bacillus cereus, enumeration of Enterobacteriaceae, enumeration of E. coli, enumeration of Vibrio cholerae, and enumeration of Vibrio parahemolyticus. So any person might be getting doubt how many counts we will do in each test. We will do all the counting, or we will do specific. Depends on the limits which I have shared just now. So each sample sometimes is having 3 tests, sometimes is having 5 tests based on the limit set by the GSO, which in turn it's coming from Codex Alimentarius. Detection test. Here, detection test is concerned only to detect, not to count. And the procedure for detection is very lengthy. Around 1 week time is there to get the final result. So detection test, the permissible level is 0 for all these organisms: Salmonella, Listeria, monophytins, and detection of E. coli, O157, H7, detection of Campylobacter jejuni, and detection of Coronobacter sakazaki. Here, I go back to the previous slide just to show you the permissible level of Salmonella and E. coli, O157. If you can see in the raw meat, 0. E. coli, O157, 0. Come down fresh poultry, the Salmonella, Campylobacter jejuni is 0. Salmonella is 1, but there is a star mark there. It is for a different type of food. Then raw minced food, you can see Salmonella 0, E. coli 1570. So generally all detection organisms are permissible level is 0. Even one organism is detected, the food is unsafe for the consumption of human. Now the methodology for the processing of food sample. Here, the sample preparation, once we prepare the sample, in the next slide, I will show you how we are preparing sample. But after a sample is prepared, we have these steps. We have to do cultural methods followed by biochemical methods and serological methods, molecular methods. So based on the type of laboratory, some laboratories go for serological methods, some labs are going molecular methods, some labs are going for biochemical methods. All the three methods are accepted by US FDA. So based on the type of laboratory and their budget, they will plan the different methods to follow. Now, this is the general stages given by US FDA, and the same steps also mentioned in ISO and GSO standards. The first step, we all know that microbiology work is very sensitive, and we should never open the sample in any other area except under aseptic conditions in the laminar airflow. And there are certain rules to receive the sample. Here, I don't have enough time to discuss this, but some points I will highlight here. Before receiving the sample, there are some specifications for microbiology. The sample should not be having any leakage or any holes, or should not be opened by anyone. Because the food diagnosis laboratories, there are two sections, microbial section and chemical section, and sample is same. So it is always advisable the first sample should be used by the microbiology analysis. So they will open the sample under sterile conditions, and the remaining part of the sample will be given to a chemical analysis. Because if chemical analysis are first opening the sample, can lead to contamination and invalid results. So that's why we should open the sample under aseptic conditions. Then we have sample preparation or first dilution. Then we have second dilution. After this, we have inoculation of the sample based on the type of pathogen, as we have discussed many pathogens there. And after we inoculate, here the inoculation methods are like generally given what we follow in the microbiology labs, spread plate and pour plate. So the incubation temperature is different here. You can see I have quoted three temperatures here: 37 degrees centigrade for bacteria, 44.5 degrees for thermotolerant coliforms, and also for the enrichment step for some of the pathogens like Salmonella and E. coli 157. And 25 degrees is for yeast and mold. Here, the time of incubation also varies, as you can see there. 24 hours to 48 hours is for bacteria, and yeast and mold is 5 days. After we get the count, we should record the results as CFU for fungi and bacterial detection. And for identification of food pathogen, there is another procedure. We have to go for enrichment. Here, just want to show the sample preparation, because in the previous slide details are not given. For enumeration tests like aerobic plate count and yeast and mold, all the international standards, including US FDA, are specifying taking 25 grams of sample, and peptone water is generally considered as diluent here. Sterile peptone water 225 ml is taken, and there is an instrument called stomacher in which we will mix the food sample, and it is having an RPM of 230 RPM for 1 minute. This is considered as sample preparation, also considered as first dilution, because the ratio is 1:10 here. 25 gram sample to 25 ml. Here, because the shortage of time, we should see that in the next one from the peptone water, you have another buffered peptone water. Then third one, you can see alkaline peptone water. These are for different organisms, but the general diluent is peptone water. Buffer peptone water is for enterobacteria, and alkaline peptone water is for Vibrio parahemolytica. So the sample preparation is different for the type of bacteria. For enumeration test, it is common, like aerobic plate count and total count of yeast and mold, we follow the sample preparation. But for detection test, we have to prepare in a different media according to the type of pathogen. Here I am trying to highlight the detection tests. On the left side, you can see the Salmonella, Campylobacter jejuni, Listeria, 0157, E. coli, and Chronobacter sakazaki. And here is the procedure. In brief, I would like to highlight here. After we prepare the sample with 25 grams, this is considered as primary enrichment. And this sample preparation is done in generally sterile packets, because the instrument stomacher is designed in such a way you have to insert the sterile packet inside and the stomacher will mix the sample. So this packet, after mixing, we go for serial dilution and the remaining volume will be incubated, and that's why it is called as primary enrichment. So what is happening here is primary enrichment. We are trying to enrich the organism of interest and suppress the growth of other organisms. Because the food sample is having numerous organisms, we want to detect our target organism. So we will suppress the growth of other organisms by certain special nutrients given in the media. In the coming slides, I will highlight the list of media, then you will understand that there is a specific media for each organism. So this first step is called primary enrichment. Then from the primary enrichment, we take 1 ml and inoculate in 10 ml of secondary media. We have 3 options here for Salmonella: Rappaport Vacillidis broth, tetrathionate broth, and fluid selenite and cystine broth. Any one we can select here. This is called secondary enrichment. Then coming to the Campylobacter, you can see first step is same, 25 grams, but the second step, the 10 ml of the media is getting changed. For Salmonella, it is Rappaport Vacillidis, but for Campylobacter jejuni, it is bolt-on broth or pressed-on broth. Again, in the case of Listeria, you can see the second stage, the 10 ml media is getting changed, Fraser broth. Then for E. coli 0157, again, it is getting changed, modified tryptophan soya broth with no biocin. And for Chronobacter sakazaki, getting changed into EE broth. So based on the type of pathogen, we have to use different media here for detection tests. Now, this is a list to show you what are the different types of media used by the food microbiology laboratory for detection test. For Salmonella, you can see buffered peptone water. There is big list for Salmonella. We have different types of media here, and this media is in different stages. We have one media in primary stage, primary enrichment, second media, secondary stage. Then for isolation, we have hectone enteric agar or XLD agar. After this, we will go for biochemical test. After biochemical test, we can go for PCR or ELISA for more confirmation. And Staphylococcus aureus, there are some media you can see. I have given a different color to highlight each different type of media for a different organism. The list is very big here. Enterobacteriaceae, Campylobacter jejuni, Vibrio, Clostridium perfringens, Chronobacter sakazaki. I would like to discuss about the E. coli and E. coli 0157. These two are similar organisms, so it is not possible to identify them based on biochemical tests. So we have to go for PCR to identify E. coli 0157 because it is having same biochemical characters like a normal E. coli. So it's very difficult to identify E. coli 157 by biochemical test. We have to go for PCR. Still, the commercial testing system has not developed to invent any new biochemical system for E. coli 0157. So we rely on PCR here. Then these are the methods for confirmation. Once we have grown the organism and we have isolated the organism, the methods which I am highlighting here is for detection organism. Because for enumeration test, the stopping point is counting the colonies and giving a count. For detection test, we have to confirm whether the Salmonella is present in sample or not, or E. coli 157, or Chronobacter sakazaki, or any detection organism is present or not. So we have biochemical methods. We can use API from Biomirex, or we can use Vitek2. It is an automated system. The Vitek2 is having two advantages. It is not only identifying the bacterial species, also giving you antibiotic resistance. Serological methods, we can use ELISA, semi-automated or fully automated. We can use FIDAS. VIDAS is a system which is designed according for the food diagnosis based on immunoassay system. The VIDAS is a limited system. It can identify only 5 organisms which I have enlisted below. It is a customized system for food diagnosis, not commonly used for other labs. we can identify Listeria, Campylobacter, Salmonella, E. coli 157, and Staphylococcal enterotoxins. Then here I want to highlight about the Biomirex solutions. They are specially designed for food diagnosis, and they have excelled, and they have improved from time to time. The basic version is the API biochemical test, where we have around 15 to 20 tests there. After we isolate the bacteria, we have to make a liquid suspension and inoculate in 20 different media and go for incubation. This is a manual method. Then after an incubation, we add the reagent and looking for the color reaction. With us just now, as I have discussed, it is a system designed for only 5 pathogens. Then Vitek 2 is having automated system. Then the latest one, most of you might be knowing, is the MALDI-TOF. So Vitek also has developed their own MALDI-TOF by the name Vitek MS. This is based on the peptide fingerprint library. So this is basically like mass spectrometric analysis, and it has a library of 15,000 microbial strains. There is a limitation here that if the organism library is not available for a particular species, then we cannot identify. With Vitek 2, we can identify more than 350 bacterial yeast strains. So for food diagnosis, the Biomirex has designed very good technology for identification. Then the other molecular methods include PCR. You can do real-time PCR or multiplex, then 16S rRNA or 18S rRNA sequencing. MALDI-TOF we have discussed just now, but there is a limitation that new isolates, unless their peptide library is not available, then we cannot identify by MALDI-TOF. Here, what I want to discuss that based on the type of laboratory, they will follow the molecular method, which method they can use. But there is a limitation here, even though there is advancement and we have molecular methods, but they all depend on the isolation of the organism till getting a pure colony. There is no method here which can directly start from the raw material. Whether you use PCR or 16S rRNA or MALDI-TOF, in market till now there is no method which is directly starting from a food sample and going to ELISA or PCR or 16S rRNA. All have to go for the enrichment isolation stages. This is the reason there is a time taken by the diagnostic labs to give the report, because microbiology confirmation of this species is taking time. We cannot straight away start with the PCR from the food sample like meat, fruit, vegetable, cooked food, rice. They all need preparation, isolation of the organism, then selective isolation, pointing towards a single colony, which is pure culture. Then we can go for these systems. So still people are working out to develop any technology so that we can directly start from a raw material, a food sample, to go for molecular methods. So this is the reason there is a time taken by the laboratories to give the report. For example, the Clostridium perfringens is taking more than 20 days to give report. Whereas the Salmonella and E. coli 157 are taking 7 to 10 days. So this is one of the limitation for the microbiology lab regarding the turnaround time for the give reports. Now coming towards the water analysis, we have the test called coliform count. These are different tests and different organisms what are tested in water analysis. Coliform count, detection of E. coli, detection and enumeration of Pseudomonas aeruginosa, detection of fecal streptococci, and detection and enumeration of sulfide-reducing Clostridia, enumeration of heterotrophic bacteria. Out of these 6, the Pseudomonas aeruginosa is followed by filtration method of 100 ml water sample, and fecal streptococci also filtration method, and sulfide-reducing Clostridia is an anaerobic method. These are the different media for water analysis. Previously, we have seen for food analysis. This is for water analysis. For coliforms and thermotolerant coliforms, the first stage media is lauryl tryptose broth. Then there is an option, modified lauryl tryptose broth or McConaughey's broth. Then in the second stage, we have brilliant green bile broth. And in the next stage, we have EC broth. For Clostridium perfringens, we have perfringens agar or reinforced Clostridial agar and cooked meat media. For Pseudomonas aeruginosa, of course, cetrimide agar is a selective media. These are the media for other organisms, fecal streptococci and heterotrophic bacteria. And for E. coli, we have EMB and TBX chromogenic agar. Coming towards the methods for water analysis, the traditional methods include the MPN, which is very popular among the microbiologists. They all know about the MPN, most probable number. We have a 3-tube method here, 5-tube or 10-tube. Also have a 15-tube method. Then for Pseudomonas and fecal streptococci, there is membrane filtration technique for 100 ml of sample passing through 0.45 sterile filter paper. Here, with respect to water, also I want to discuss about the industries. They not only test the water, drinking water or water for human consumption, but there is a routine water testing for the plants, your treatment plant, and the water used in the formulation and production. For these also we have to do E. coli, membrane filtration, Pseudomonas and other tests. And for sulfide reducing Clostridia, there is an anaerobic culture technique. These are the routine methods, but here I am discussing one advanced method called quantitative system. It is given by IDEX company. So this is similar. This is based on actually most probable number, but it is a advanced system. There is, in the case of MPN system here, 3-tube, 5-tube, 10-tube, you will be taking different volumes of the water here, and you will be taking series of tubes with Durham tubes. But the quantitray is an advanced system. There is a ready-made media provided here called Colisert. We will see in the next slide. And the 100 ml water sample is directly mixed with the reagent. And there is a tray here consisting of 51, tubes. So we will be adding sample 100 ml water. And after incubation, we have to count the number of positive tubes and refer the MPN table. This is the procedure how we will do the quantity. First step, we mix the water with the reagent, Colilert reagent. Then we will seal the tray, and then we incubate the tray and count the number of positive tubes here. Wells, not tubes, wells, sorry. And the broad procedure, briefly, I have given in a box here. 100 ml of sample is divided into 51 wells, and then it is sealed and incubated. And there is a separate MPN table standardized here for quantitray. The normal MPN table is for a manual method, what we do 5-tube, 3-tube, or 10-tube method. But here for quantitray, they have a separate MPN table. This is the diagnosis of food and water, which we are doing in our laboratory here based on GSO, ISO, and FDA standards. If anybody is having doubt, they can ask me.
Yeah, thank you very much, Dr. Ali, sir. That was really very informative lecture on foodborne bacteria and their diagnosis and testing methods. First of all, I would like to thank our President Arvind Deshmukh sir for creating a platform for global exchange of research and expertise. And on behalf of Microbiologist Society India, really thank you very much for the lecture. And now it's open for any questions. If you have any questions, please now you can ask any questions.
If anyone is having queries, they can email me.
Sujata. Hello.
Yeah, good evening, sir.
Good evening. Good evening.
Yeah,
I'm traveling.
Yes, sir.
Yeah.
Yes, sir. Yeah,
But
I got the
even
message.
then, yeah,
Yeah,
I'm
thank you very
traveling, but
much,
even then,
yes, sir.
yes, I'm really
Thank you very much
impressed.
for making some time, sir.
Correct. I'm really impressed by the lecture. And he is talking from Saudi Arabia.
Yes, sir.
I want to assure him that this lecture is not the end.
Yes, sure.
It is the beginning of, and we have to do many things in the future. Dr. Swarnaji Singh, Dr. Swarnaji, so attending
Hello,
your
sir.
lecture.
Hello, sir.
Yes.
Hello, sir. I am Dr. Swaranjit here.
Along with the yes. Thank you.
Hello, I am Dr. Swaranjit here.
Yes, doctor.
Can you hear me?
Yes, I can hear you.
Very good. It was an excellent lecture, as Dr. Deshmukh said. I really enjoyed the lecture. Now, I want to ask you a small query.
Yes.
For microaerophilic microbes,
Yes.
in your case, you mentioned about Campylobacter.
Yes. Campylobacter jejuni, yes.
Yes, usually. So how do you culture them from the water or food?
Yes. Thanks for asking this question, because I have not discussed in the presentation. We actually, in my laboratory, we have a anaerobic system from US. There are two things here. We need to provide a CO2 incubator and with 10% carbon dioxide. And there is an anaerobic system which is connected with a mixture of gas, nitrogen, oxygen, and hydrogen. So based on the metabolic requirement of the organism, we can set the ratio in this. So it is providing, there is a, to begin with this, after you prepare sample preparation and do serial dilution and plating, the plates we are keeping in an anaerobic jar. This anaerobic jar is connected to an anaerobic system. The anaerobic system is having a testing menu. We can set the percentage of hydrogen, nitrogen, and oxygen. And then it will take a time to clear and add the gases in this atmosphere, around 5 minutes or 3 minutes. After this time, we can take this jar and keep in a CO2 incubator. And CO2 incubator, we can set the percentage of CO2.
Okay. And the jars are from which company?
Yeah, I will share the. It was there in my Riyadh municipality lab. Now in our green lab, I will purchase in the coming days, because it's a private lab, so there is some budget constraint. But in my previous lab in Riyadh municipality, I was having 2 anaerobic systems and 2 CO2 incubators. This product is from USA. I will be sharing details with the madam. You can take from her.
Yeah, because you having the jars in the private company is very important.
Yes.
Because you have to see the microaerophilic and maybe anaerobic cultures also.
Yes. I was using this for 2 organisms, for food Campylobacter jejuni, and for water for Clostridium.
Okay, I see. Because they require completely anaerobic atmosphere.
Yes.
So you can
I will
make
share the details. At the moment, not recollecting the name of the company, but I will share the details with the madam. She is in touch with in WhatsApp, and you can take with her, or I can give in our group also.
Very good, very good. Okay, thank you very much. Thank you.
I'm thankful for the Microbiology Society, especially our president, Dr. Arvind Deshmukh sir, and Dr. Sujita Chaitanya ma'am, for giving a chance to share our knowledge.
Yeah.
Thank you so much.
Thank you.
Thank you very much, sir. Thank you everyone for attending the lecture. Thank you very much. With this, we close the session. Thank you once again. Thank you very much.